call. Am I audible to you? Hello. Okay, I'll share my screen. Then we can start with our today's session. I hope my slides are visible.
Yes, ma'am, it is visible.
Okay, thank you. So we'll start with our session. So today we'll be having a problem solving session for week 8. So the topic that we will be covering are the biases in RNA-seq experiments, data normalization method 1 and 2, part 1 and 2, differential gene expression analysis, and differential gene expression analysis results and visualization. So again, it will this session again will have questions. So first, I would like you to try to answer those questions. Then we will look into the concept with going ahead. So just try to answer the questions first. Then we will look into the concept for each questions. So I'll briefly introduce myself. Oh, I am Pooja Yadav. I am a PhD scholar and a PMRF fellow in IIT Bombay, in Department of Biosciences and Bioengineering, and I am a PhD student in Molecular Virology lab. So today in today's session, so till last week we have covered the analysis of quantification of RNA-seq data. Now we need to analyze our data that we have got, just to see whether the data that we are getting is I mean, if we have a two type of samples, one is disease sample, one is a normal sample, and we extract their RNA from each sample, then we have to analyze what gene is expressing more or less in order to give that particular disease phenotype. So for that, there are few normalization and analysis technique that we will be discussing today in today's lecture. So today's lecture questions will be based on certain normalize RNA-seq normalization data and how to analyze those data. So in question number 1, what is given? We are given with three genes with their length, and we have the read count data from three replicates. So this is from the same sample, but we have a three replicates of the same sample, and after the sequencing, we got these read counts from each experiment. So in this question, what is asked? What will be the read count of gene C in replicate 2 after TPM conversion? TPM is transcript per kilo base million. So please try to solve this question. Take few minutes and try to answer this question. Then we will look how we can solve this question. So we are given with the raw reads. We have to normalize our data so that we can analyze our data. And here TPM normalization has been given. So you need to convert your data as per transcript per kilo base million. So this is basically used when we normalize our data within the sample. So here are the one sample, and for a sample, three replicates are given for each gene. So basically, there are three steps in TPM conversion. First, we divide the count by the gene length. Then we estimate the scale factor. So try to like divide these count by its gene length and try to solve this question first. Okay, let's look into the concept first. So there are basically three steps in TPM conversion. So this is our data that we are given with. So first we need to divide the count by the gene length. So here are the gene length given. For A it's 1 kb, for B it's 2 kb, for C it's 5 kb. We divide our these raw reads with our gene length each. Like you have to divide this by its gene length, from replicate to 17 by its gene length, that is 1, then 3 divided by 1. For it you need to divide 8 divided by 2, 11 divided by 2, 6 divided by 2. Like this you'll have to divide for each gene length. You have to divide read count by its gene length. So after dividing you'll get this table. Then you have to estimate the scale factor and then divide by million. So how you get the scale factor? So you for the scale factor, so in the question it is, we are asked what will be the read counts for gene C in replicate 2. First, how do we calculate the scale factor? We add all the gene count uh, for that specific replicate. So in this question it's replicate 2, so we will add its all the reads that we get after dividing its gene length. So it will give the scale factor. Then we will divide it by a million. So after getting a scale factor and we divided it by a million, we get 22.7 into 10 raised to power minus 6. So now we need to calculate the TPM value for gene C of replicate 2. So for gene C, uh, after dividing its length, we get this number, that is 0.2. So we need to divide this number with our scale factor. So that will give us the TPM value, which is transcript per kilo per kilo base million. So I'll just uh, repeat this again so it will be more clear. So we are given with the sample. There is a one sample, and for a sample we are given with three experiment conditions which are replicates of the sample. And here we are analyzing three genes. We are given with the raw reads of each replicates. So to calculate TPM, first we will be dividing each raw read with the gene length. So let's suppose we are dividing this 10 by 1, 17 by 1, 3 by 1, 8 by 2. Then we'll get a table. From here we need to find the scale factor. How do we find the scale factor? Simply we need to add all the new values for each replicates. So when we will add for replicate 2, we'll add all the values and divide it by a million. We'll get a scale factor. So after getting a scale factor, to calculate TPM, we'll have to divide our new value that we get after dividing by its length by the scale factor. Then it will give the TPM value. For this question, the TPM value would be 8810.57. So the correct option would be, so you can give a range between 8810 to 8811. So but the the exact value is 8810.7 .573. Is there any doubt in this question? So again, this type of conversion or a normalizing method is used within the sample. So we have a one sample, we have a replicate of that one sample that we give, we to normalize the sam the this the count, the read counts, we use this TPM conversion. This is one of the normalization method. Okay, if there are no doubt, we'll move to our next question. Question number 2, it states that gene A counts in four different samples. Gene Gene A counts in four different samples are 1000, 270, 156, 190 respectively. What will be the pseudo reference sample counting according to DEC2 normalization process? The pseudo reference sample is generated by taking geometric mean. So here the gene count of four different samples are given. The gene A count, there is a one. So what happening here is we have a four sample, and for this four sample we are analyzing for a gene, which is gene A in this case. So for each sample, the read counts for gene A is given, which is 1000, 270, 156 and 190. So here what they are doing, they are DEC2 normalization. So for that you need to find a pseudo reference of the sample. So it is already given that how do you find the pseudo reference sample. So pseudo reference sample is generated by taking a geometric mean. So basically you need to simply find the geometric mean of all the values given. So please try to calculate the geometric mean of the value, so you'll get the pseudo reference for this normalization method. So geometric mean is simply the root value of the multiplication of these values. Okay, so let's look into the concept. How do we calculate the pseudo reference sample? So we are given with a gene A count in a four different samples that are 1000, 270, 156 and 190. So the pseudo reference sample will be the geometric mean of these values. So we first need to multiply all the value, then we'll have to find the root of that value. So after the geometric mean, you get the value to 199.096. That would be our pseudo reference sample size. So since there are different gene count given for different samples, so here we need, for this normalization method, so we are first trying to find a pseudo reference sample size. So like we can compare or we can normalize based on this pseudo reference sample size. So that for that we need to take the geometric mean, and if we take the geometric mean of these value, we'll get 299.096. So in this you can give a range between 299 to 300, but the correct value is 299.096. So I hope this question is clear. Okay, let's move to our next question. Which of the following model is more suitable to handle overspreaded data? First is Poisson, second is binomials, third is negative binomial, fourth is exponential. So for overspreaded data, it means the variance is like it's greater than the mean value. So so for these type of data, what should be the model that we should compare our value in? Like what sort of model it should follow, um, or we should use to handle these overspreaded data? Please try to answer this question. Okay, I'll share you the link. So the basically the answer is negative binomial. So I'll share you the link. Please go through the link. So this, the negative binomial regression or a negative binomial, it can be used for overspreaded count data. That is when the conditional variance exceed the conditional mean. So so here is our mean for negative binomial. So this is our mean, and you can see that the variance in our data is exceeding the mean. So like this data is quite overspreaded. So in binomial, what happens is this curve follow like this, the bell shaped one. But for the negative binomial, so you have this negative axis as well, the curve would follow such, like, and it will spread uh, at the x-axis. So to this, so it is used basically when our, like, used over overspreaded over dispersed count data. So basically when the mean, the variance is exceeding to the mean. So here you can see that for the negative binomial, this axis would be spreaded, and you will see a lot of data, count data here. While in case of just binomial, you just see this bell curve, and it will, like, it won't spread on the x-axis, but for negative binomial, it will a lot spreaded on the x-axis. So the answer would be a negative binomial. So to handle the over dispersed data where the data count is far like apart from the mean values, so we use these negative binomial model. So you can go through this link for a minute just to know more about it, or you can go to this link after the class, so you'll get to know more about it and it will be more clear. So let's move to our next question, question 4th. Um, in which normalization technique m log fold change with respect to reference and a average expression with respect to reference, values of each gene across every sample is calculated and it trims 10 to 20% of m and a values. A option is TPM, B option is TMM, C option is median normalization, D option is upper quartile normalization. So in this question, this is one of the normalization. We plot m and a value. M is log fold change with respect to reference. A is average expression with respect to reference. Then we trim 10 to 20% of the m and a value, and then we plot this m and a. So in what sort of normalization method this sort of analysis is used or done? Okay, so for this I'll share you a link. Please go through this link, go through this manual. So for TMM normalization, we plot M and A, M versus A. M is log fold change with reference to with respect to the reference, while A is the expression, gene expression with respect to reference. So in TMM normalization method, we calculate these change in our signal or the data we get, and then we plot with respect to the reference, and then we further analyze our data if there any cluster is forming, or if there how our data is spreaded, or how there is, like, we compare the fold change with respect to the number. Fold change basically the count, read count number with respect to the expression of our gene of interest or between the samples. So these sort of normalization methods, in the initial question, those sort of normalization method was done within the sample, and these methods are used between the samples. So for this uh, they managed to overcome many biases like RNA-seq count biases or specific sequencing preference biases, GC count biases, etc. within the sample when we compare within the samples. So just go through the link. So the correct option would be TMM normalization technique, which is option B. Let's move to our next question. Okay, so here we are given with read count for different sample for different genes. So in the question what is asked, what will be the size factor of sample A according to DE-seq2 normalization process? So it is the extension of the question number 2 where we found the pseudo reference sample size. So it is the continuation of that question. So in this what is asked, what will be the size factor of sample A according to DE-seq2 normalization process? Please try to answer this question. So uh, we calculated the pseudo reference uh, pseudo reference count. So first calculate the pseudo reference count for this, and then you have to calculate the size factor. Please try to solve this question. Can anyone answer this question? Okay, let's look into the concept for this question. So for DE-seq2 normalization, we count the pseudo reference count. And how do we count the pseudo reference? So we take the geometric mean for the read counts from each sample for a particular gene. So after calculating the geometric mean for these value, you get 26.57 for gene A. For gene B, you get 69.74, and for gene C, you get 76.22. So basically, these values are the geometric mean of the read counts from different sample for a particular gene. So you just need to find the root value of the multiplication of these values. So after, and then how do we calculate the size factor which is asked in this question? So we divide these value. So these are the pseudo reference count, and we are given with the raw counts as well. So when we divide our raw counts with our pseudo reference count, we get a certain value. So for sample A, we need to calculate the size factor for sample A. So for a sample A, for gene A, it would be 25 divided by 26.57. For B, it would be 105 divided by 69.74. For C, it is 90 divided by 76.22. So, so these are the values we are getting for a sample A. If we calculate the median of these values, we will get the size factor for sample B. So when we calculate the median of these values, we'll get 1.81. So this will serve as the size factor for sample B. Sorry, for sample A. So here, instead of taking different values, we are calculating a one values for all the samples, for all the genes, so that we can compare with a different sample. So in a sort of, we are normalizing our data. So basically what we are doing is we are normalizing our data so that we can compare, we can neglect we can, I mean, discard, we are not discarding, but we are like, uh, we we are discarding these biological, we just want to have a biological variance, but we don't want to have a technical variance. What the variance we are getting in the expression is only coming from the biological variance. So to find the only the biological variance, we for to like analyze the data, first we need to normalize our data so that we can like discard these technical variants, so that we can uh, from our analysis we can discard these technical variants that might be coming from a sequencing or error or sequencing biases or something else. So we normalize our data so that we, if when we are getting a result, we can be very sure that these results are coming only from the biological variants, not from the technical variants. So for this we are calculating the median value, which would come as 1.181. So for this question you can give range between 1.10 to 1.25, but the exact value is 1.181. I hope this question is clear. Let's move to our next question. What is type 1 error? A, true positive, B, false positive, C, true negative, D, false negative. Please try to answer this question. Okay, let's look into the concept. So let's suppose we have a patient. So from the test, some pathological test uh, of the patient. So when we test them pathologically, we get their real condition, whether they are diseased or not diseased. So we get either they are positive for the disease or negative for the disease, or uh, and from our findings, when we are analyzing our RNA-seq data, this is our study finding. So this is the real value and this is the observed value. You can say that. So when the real value is also positive and what we are getting is also positive, the both the values are positive, we call it as a true positive. That what we are observing is also what we are expecting. So the expected value is same as observed value. But let's suppose after a test we get to know that the patient is negative, but our analysis or our sequencing data, from our sequencing data, the sample is coming out to be positive for a disease. So from our finding, this expected value is different from the observed value. The observed value is a negative, while the expected sorry the observed value is positive while the expected value is negative. So this turns out to be a false positive because we are observing it as positive, but the reality is negative. So we call it as a false positive. What we are getting is false, we are getting positive, which is false, it is false positive. So it is coming under type 1 error. So, and if our observed value is positive and if our expected value is positive and what we are observing is negative, which comes out to be false negative, because the reality is positive and we are getting a negative, this is false negative. So it is type 2 error, and if both are negative, it is again a true negative. So true positive, true negative, apart from these two conditions, false positive or false negative, we should not rely on that. This is wrong. So for the false positive, if the observed value is negative, if the expected value is negative but the observed value is positive, it would come as a false positive and it comes under a type 1 error, which is false positive. So the correct answer would be B. The what is type 1 error? It is a false positive. I hope this question is clear. We'll move to our next question. Question 7. Total number of genes that are used in a differential gene expression analysis is 20,000. How many genes are expected to be falsely positive if the FDR, false discovery rate, is said to be less than 0.05 and the total number of differentially expressed gene is 500? Please try to solve this question and try to answer this question. So, so in the experiment, 20,000 genes are analyzed, out of which they have found 500 genes to be differentially expressed, but their false discovery rate is said to be less than 0.05. So basically this percentage or this fraction can be a false negative, a false positive of what we are observing. So based on this information, try to calculate this answer for this question. So basically we need to calculate how many genes can be false positive from the results that we are getting for differentially expressed genes. Can anyone try to answer this question? Okay, let's look into the solution for this question. So, in this question, we had 20,000 genes, out of which we need to find how many genes are differentially expressed. So, after the experiment, we have found that 500 genes were found to be differentially expressed, but the false discovery rate is less than 0.005. So, this fraction of the gene can be falsely positive, what we have found. So, the expected number of a genes that could be false positive is 500 into 0.005, or the 5% of this number can are the genes that can be false positive. So when we multiply with our differentially expressed gene with our false discovery rate, we get 25. So the correct answer for this question would be B, which is 25. I hope this question is clear. Let's move to our next question, question number 8. The introduction of spike in sequences in the count data can help in better normalization, removing technical artifacts and variation introduced during experimental setups from the data, estimating read counts in each cell, identifying doublets. Please try to answer this question. So, spike in sequences are basically the known quantities of RNA then we add to our experiment. So, basically, why we add these RNA from our side? Why, what they can help us? How can they help us to like analyze our data more efficiently? Please try to answer this question. So when we add these RNA from our side, so any variation that we are getting must be coming from the biological variation, not from the technical artifact. So we know how much or the quantity of RNA that we are adding, and then when we see the difference after we add the known quantity, and whatever the difference is coming, it is coming from the biological variations. So the spike in sequences in the count data, they can help in better normalization, or also they can help in removing technical artifacts and variation introduced during experimental setups from the data, because we already know that what we are, how much RNAs we are adding. So any variations that are coming is coming from the biological variation, not from the technical variation, because we are already covering the technical variation from our end. So what we know that what variation we are adding. So it will help us to better normalization, because we already know like this is how much we have added, so the difference is coming from the biological variations, and then we are removing the technical artifacts and variation introducing during experimental setups for our data. So the correct answer for this question would be, okay, so spike in sequencing, they are known quantities of RNA that are added to each samples in experiment. Since they have constant concentration across all the samples, they serves as a reference point of normalization, that as I've said. So introducing a spike in sequence can also help to identify and account for technical variation introduced during the sample processing and sequencing. So again, the it can help in better normalization as well as identify and account for technical variations. So the correct options would be A and B. I hope this question is clear. Let's move to our next question. In an NGS data file, it was observed that a particular region, the coverage in a disease sample was 40, whereas the coverage in a healthy sample was 80. The total number of read present in the diseased and healthy sample was 2000 and 4000. Calculate the coverage ratio. So we are given with the coverage as well as the total number of reads for healthy, for the healthy sample as well as the disease sample. So we need to calculate their coverage ratio. Please try to calculate and try to answer this question. Has anyone solved this question? Okay, so how do we calculate the coverage ratio? So to calculate the coverage ratio, so so we divide the coverage ratio in disease sample divided by total number of reads in disease sample divided by coverage ratio in healthy sample with total number of reads in healthy sample. So the coverage for the disease sample is given 40, while the total number of reads are 2000 for disease sample, and the coverage for healthy sample is given as 80, while the read for healthy sample is given 4000. When we divide this value to this value, we get 1:1 ratio. So the correct option would be C, 1:1 for this question. So I hope this question is clear. So we'll move to our next question. So in this question, we are given with 3 samples. For each sample, we are given with the read counts of 5 genes A, B, C, D, E. So in this question, what is asked? What will be the value of gene B after counts per million normalization? So after CPM normalization, we need to find the value of gene B in sample B. So please try to calculate for this question.
Zero point 0.3 into 10 power 6.
0.3 0.3 0.3 into 10 raised to power 6 is incorrect answer. Can you retry? Okay, let's look how we can calculate the CPM for gene B in sample B. So to calculate the CPM value, so first we need to add all the read count for all the genes. So in this question we are in this question we need to calculate for sample B. If we add all the values for all the genes of sample B, we get 65. So for how to calculate the CPM, so then we divide our particular gene of interest. So in this question we are asked for the gene B from sample B, and we divide the value that we got after addition of for all the genes from a particular sample. We divide for particular our gene of interest. For a gene B, it is 6, so we divide 6 by 65 and then we multiply by 10 raised to power 6, which is a million. So when we solve this 6 divided by 6 into a million, we get 0.09 into 10 raised to power 6. So the correct answer would be, so you can give a range between 92300 to 92310, but the correct answer would be 92307.69. So let's move to our next question, question number 11. In differential gene expression analysis, the volcano plot shows the relationship between statistical significance and fold change, the relationship between mean normalized count and fold change, the significant differentially expressed gene including outliers, the underlying phenomena that the method which was chosen for normalization was correct or not. So I'm sharing a link with you. Please go through the link and try to answer this question. So after normalization and for the analysis, we plot our differentially expressed gene just to analyze our data. So one such plot is volcano plot. So what information does this volcano plot give? So for the volcano plot, we plot the log 10 of p value plus the fold change value. So it is the relationship between the statistical significance and fold chain. This is correct option. And for the C option, significant differentially expressed gene including outlier. So if we are plotting the statistical significance with the fold change, we get the idea of the significantly differentially expressed gene including outlier. So it gives us the information about the genes that are differentially expressed and are significant. I mean the p value, they are statistically significant, and with this we can also comment on the outlier for the same. So the correct option would be A and C, the relationship between statistical significance and fold change. C option is significant differentially expressed gene including outliers. So let's move to our next question. Which kind of clustering method is used by heat map? So density based clustering, fuzzy clustering, hierarchical clustering, k-mean clustering. So it's a very straightforward question. The correct answer is hierarchical clustering for the heat map. So it clusters based on the same sample, so which is called the hierarchical clustering. So for the heat map, this hierarchical clustering is used, which cluster the same sample in which collect the same sample in a single cluster. So the correct option is hierarchical clustering. So let's move to our next question. Which of the below option is linear dimensionality reduction technique? Density based clustering, principal component analysis, uniform manifold approximation and projection, heat map analysis. Please try to answer this question. So we have a data of, like, we have a multiple data. Okay, our data is distributed in having a varying dimension. Like, you cannot pick one and based on that you can analyze your data. First you have to reduce your data, like, and find out a main difference among all the data that you have, and which is called the linear dimensionality reduction technique. So is uh, which technique is actually the which do this linear dimensionality reduction of our data. So let's suppose you have multiple samples, like 100 samples, and for each samples you are analyzing for 100 genes altogether. So you'll have to find a main component. So this is a multi-dimensional data, right? Because you have hundreds of gene for hundreds of sample. So there is no one dimensional dimension in the data. So it has a multi-dimension. So you need to reduce this data into a linear dimension. So that linear dimension should cover all of the major differences in each of the dimensions or each of the variations, each of the genes. So what sort of techniques which is for the linear dimensionality reduction of the data? So the technique which is used is principal component analysis. So what principal component analysis that does, that from a multi-dimensional data, it analyze the data, and uh, it extract, based on its analysis, its own analysis, it extract the one single data which covers most of the differences between the sample and the genes. So the correct options would be principal component analysis. Is there any doubt?
Uniform manifold approximation can be used, right?
Uniform, because our data is actually like a multi-dimensional data. For the uniform manifold approximation, like, it can be used, but it does not cover the most of, like, it does not used for the multi-dimensional data.
Okay.
So if you are going for a multi-dimensional, such a huge complex data, it is preferred to do the principal component analysis, so that it can get that one dimension which covers most of the variation among the data. move to our next question. Question number 14. Which underlying phenomena can we identify by visualizing an MA plot? So, please go through this question and try. So, the answer are already given in this question. So, for MA plot, this plot also helps us to understand if the input data has gone through the correct normalization steps or not. Highly differentially expressed gene or technical artifact such as outlier can be identified MA plot. So MA plot is basically we plot the fold change versus fold change with respect to the reference versus the gene expression with respect to the reference. So we can, by the MA plot, we can help to understand if the input data has gone through the correct normalization, and also the highly differentially expressed gene or the technical artifact as an outlier, they can also be identified from the MA plot. So the correct option would be C and D. So let's move to our next question. What will be the value of gene C? So okay, so again, for here we are given with the sample, where 3 replicates of a sample is given, and we are analyzing for 3 gene with a varying gene length. So in this question, what is asked? What will be the value of gene C in replicate 1 after read per kilobase per million RPKm normalization? Please try to calculate the answer for this question. So you need to do RPKm normalization here. Can anyone answer this question? look how we can calculate the RPKM value. Okay, so how we calculate the RPKM values? So first uh, we add all the values for we add all the values for gene for a particular replicate. So we need to calculate for replicate 1. So we'll add up all the values for each gene. So after adding all the values, all the read counts, we get 33 for replicate 1. So we need to calculate the RPKM value for gene C for replicate 1. Then we will divide this value with our calculated value, which is 33. Then we'll get a number. Then we multiply it with a million. And then we divide it with its gene length. So read per kilo base per million is basically including the gene length bias. So to cover up the gene length bias, like what we have done in the CPM, we do the same, but after this we divide it by the gene length. So after dividing it by gene length, we get the value of 91000. So you get the value around 909, which is around 9100. So you can like give an answer in a range between 90,000 to 92,000, but the correct value is 90909.09. So that was our last question for today's session. So if there are any doubts, we can discuss them. Just try to go through each normalization method. So what are the normalization methods between the samples, or within the samples, or between the samples? How do we normalize our data before analyzing it? So we need to be sure there is no technical biases in our sample which is hindering with our biological variance. So for that you need to normalize your data first to analyze this data. So there are different normalization method. So you need to find which is best suited method for your data, or which are commonly used, and how can you use them, how can you normalize your data before analysis. So just again revisit your lecture slides and try to, go through it again, and then you will, I think, able to understand more now, because now we have seen what sort of question that can be asked. So if you go through the lecture slides again, then you will able to connect with these and can try to answer these questions. So that was all for today's session. We'll meet next week at the same time for our problem solving session, week 9. So if there are no doubts, we can wrap up our session. So you guys can leave the meeting if there are no doubts.